I'm going to share some of the results we've been working together. So I'm going to talk about the new regulations of the use of antimicrobials in the U.S. We're going to discuss some of the data analysis we've been performing and some of our preliminary results and also the next steps in the project. So the project is a collaboration between the VPM department at the University of Minnesota with the VDL. And we're going to approach the issue of the antimicrobial resistance and the antimicrobial use. So this is the antimicrobial resistance is a trending topic that affects directly the public health, and that's why this is being discussed a lot. And there is a big pressure and an increased perception that the antimicrobial use is affecting the antimicrobial resistance. So we need to assess how it's being used in the food animal industry and how it also affects the antimicrobial, if it increases the antimicrobial resistance. So the antimicrobial use is really difficult to measure, and how it affects the resistance is also difficult to measure. A little bit of the framework of the new regulations. So the FDA released a few documents. They are the guidance for industry. And in 2017, it started the new veterinary feed directive, where the use of some drugs as growth promoters are not allowed anymore. So between 2013 and 2016, it was voluntary to adopt, and from 2017 it's mandatory. So what it affects veterinarians, producers, and pharmaceutical companies, and mainly the principles that they are suggesting is that these drugs need to be used only in clinical cases and need to be prescribed by vets. And we need to understand how it will affect the industry in the next steps. A little history. So in the EU, it was banned in 2006, and the use of antimicrobials have been, the use and the resistance of antimicrobials have been assessed in many studies, and in the Europe we have some results, but in the US it's still beginning. One of the first initiatives is NARMS, where they are assessing some results, some bacteria that is important in the industry and also in the public health approach. But we still don't have many results in how it affects animal health and animal production in the US. So the project has two objectives, and the first one is to assess the data that the VDL at the University of Minnesota has to understand how it will affect animal health and also the resistance. And so we will analyze data from 2006 to 2016 to understand what was the pattern of submissions in this period, and also how the susceptibility of clinical isolates was during this time. We chose data, only swine samples in the same time frame, so 2006 to 2016. We excluded samples that were not from the US, that had research purposes and pharmaceutical company submissions, and we kept only samples that came from diagnostic and surveillance. We had samples from 42 states, but the most common was from Minnesota. It's almost 70%. We have 36,000 submissions, but we worked with the bacterial results, so the isolates, not the isolates, the results, because it's 150,000 samples. From those, 79 were recovered from culture, bacterial culture, and from these, 29% was tested for resistance, and 20% came from PCR, and just 0.6% was from immunohistochemistry. We chose for the project only pathogens that were important in the field and also common at the VDL. And this is the list that we are working with. And the analysis started understanding the data set from that period. So we started first describing and trying to understand trends in the submissions. So the type of samples, seasonality, or anything we can find during the description time. Second step is to understand the frequency of identification of each of the pathogens in that list. We need to understand this part too. And the third step is the AMR pattern. So we chose isolates only in a farm-month level, so isolates from the same time period were excluded, so we don't have repeated information from this area. And we chose to test them for medically important drugs that are also used as growth promoters. So in one of the GFIs, there is an appendix that shows the drugs that are critically important and highly important. It's a long list. We can't discuss all of them. So for the sake of our discussion, I'm going to choose to focus more on ceftiofur, enrofloxacin, and ampicillin. So I'm going to show some of our preliminary results. We don't have, it's not all explored yet, but we have some of them, and I'm going to show. This is a graph with the overall of our isolates, the 150,000. It's a lot of information, but since we're trying to describe, to understand patterns, we need to take the big picture. So here we have the timeline, and here we have the aggregated count. This is not percentage yet because we're still in the first steps. So here in the bottom in gray we have Streptococcus suis, that is the most common pathogen. Then in green here we have Pastorella. We have E. coli in green and pink. So the purpose of a graph like this is to try to understand if there is a pattern or anything that we would like to focus more and understand. When we open this by each of the pathogens, we have the timeline for each of these graphs, and we can see that many of them are pretty constant a long time, but we have some that has decreased. So we can see Haemophilus parasuis, Pastorella, Salmonella, and Streptococcus with a decreased trend. But some showed an increase, like Mycoplasma hyopneumoniae and hyarines. Again, this is not yet a frequency because we don't have the whole data and whole analysis. This is just a picture, but it's important to identify this kind of trend to explore better our data and try to understand what this will look like in the future. So thinking about seasonality, this is a general distribution of our results along the months from January to December. We can see in general we had less results in the winter, and when we look at each of the pathogens, we can see Lawsonia, Mycoplasma, and E. coli, they could have a different pattern. So we need to understand and explore better this to see how it's going to change over time. General pattern of specimens, so most of them come from lower respiratory tract, followed by enteric samples and systemic samples. Animal category, almost 35% are undetermined, so we don't have information of age, which is a limitation, but we need to deal with this kind of data. But with the ones that we have the information, we can see that the most common is nursery, 29%, followed by growing and finishing. So some of the results regarding susceptibility I'm going to show. Just remember, just highlighting here that I'm going to talk in percentage of resistance in each of the slides, and that we're working with farm month level isolates, so we don't have that many. We have less because we filtered a little bit. When we look at the temporal trends, first in Salmonella and our timeline here, we can see different drugs that we tested and how the percentage of resistance progressed along our time. It's a lot of information together, but the one that we can see easily that changed is the red line here, that is enrofloxacin. E. coli had the same trend, so we can see that many of them are pretty constant, but we have a red line that is increasing. When we put them together, I cleaned everything that was not the focus of our discussion and kept only ampicillin, ceftiofur, and enrofloxacin. We can see in yellow here ampicillin, ceftiofur, and the increase of enrofloxacin. Same pattern for Salmonella and E. coli. If we try to look at those isolates from the point of view of pathogenicity, I try to create here a way of seeing this better. In colors, we have potentially pathogenic Salmonella, and monochromatic colors here, that potentially not pathogenic. And we can see that there is a difference for each of the drugs tested here, right? So it's different, the percentage of resistance for pathogenic and non-pathogenic. When we put E. coli together, again, same idea. So in colors, we have the virulence factors positive isolates compared with the non-positive ones. And we can see again that there is a difference between some of them. When we choose to discuss the groups that we chose, ampicillin, ceftiofur, and enro, we can see also that there is a difference. So this is something we need to explore a little bit better. If we use a different approach, this is a result of isolates that were resistant to enrofloxacin, and we used the machine learning approach to understand association with other factors in those isolates. So between those, we tested this list of predictors, and machine learning is a way of ranking variable importance. And here we can see that besides being resistant to enrofloxacin, it also shows difference with other factors. So here in this graph, this dot here, up here, is the most important, the first one most important, followed by this, and there it goes into the end. And if we highlight the four first steps, we can see that there is an association between being resistant to enro to also having the toxin STA, being also resistant to sulfa, and Fimbria F4 and LT2. So this is important to understand other factors that can be associated with resistance. And if we change from enteric to respiratory, and we see that, if we remember that in the data set, the most common pathogens that we recovered, Streptosuis and Pasteurella multocida were the most common. And if we compare the trends in the timeline for each of them, we can see that they are constant for these 5 drugs that were analyzed. And we don't see the same trend that we saw for enrofloxacin, in example. So there is a difference between those two, and this is one of the trends that we need to understand better. So to conclude, we have data. We have a lot of data. 150,000 isolates and results is a lot. And we need to use this kind of data to understand how the new regulations can affect the industry and the evolution of antimicrobial resistance. We're looking into 11 years of data. The patterns were not constant. There is a difference in time. There is a difference between pathogens. We observed that there is an increase in enrofloxacin resistance, and this is something important because it's in the list as critically important. And our further investigation will use this 11 years of data to compare with 2018-2019. So we're using this first period to analyze the past and compare with the future that is already happening now. And we had trends in AMR that are detectable, and in our preliminary results, it's different between respiratory and enteric, and we want to understand this better. And when we look at these drugs, there is a difference that we want to understand. Our next steps is first, determine the frequency of isolation. Because, like I said, we still don't have the whole picture. We're still working on that. And we need the denominators, and we need the post to compare both periods. And the other next step is to complete the analysis with the other bacteria. So it's a lot of isolates, a long list of results and pathogens that we're using. And we don't have that. We're not showing, and we don't have everything yet. And in the final step, we're going to build a statistical model to analyze the strains and understand what will happen after 2017 when the new regulation started. And with that, I would like to thank Dr. Fabio Vanucci for the technical support with the project and also for the invitation for me to be here today. The STEMA group that is always supportive with everything, and my advisor, Dr. Julio Alvarez, also for the support and all the learning process here. And thanks everyone for being here today.